Good evening to all. At the outset, I'd like to thank Dr. Sudeep for giving me this opportunity to share our recently published article on identification and validation of plasma biomarkers for diagnosis of breast cancer in South Asian women, which was published in Scientific Reports last Friday. The schema of the work which has been done is given below. So here we started off doing gene expression study of breast cancers using microarray technology. And for this we used apparently normal tissues, paired normal and invasive tumors. Apparently normal are samples which are obtained from patients who underwent surgery for benign breast condition, and these samples were taken well away from the lesion and histopathologically confirmed to be normal. Paired normal are samples taken from the breast which had the tumor but well away from the tumor and again histopathologically confirmed to be morphologically normal with no tumor cells. And then of course we had the invasive tumors. We had over 1800 differentially expressed genes detected, and from this we then selected 60 genes for validation using quantitative real-time PCR. Once we had the validation done, we then selected 15 of these genes which were secreted or potentially secreted for further analysis at the protein level. And these 15 genes were also run or checked out using geomicroarray dataset which had 176 primary breast tumors and 10 normal breast samples. We started off doing the conformation differential expression first in the tissues and then in the plasma. And for this, we used the quantibody array, which is a multiplex sandwich ELISA on glass slide. Now, we ran breast tissue lysates, and these consisted of 6 apparent normal, 23 paired normal, and 46 tumor samples. And 9 of the 15 proteins were found to be differentially expressed in the tissues and concordant with what we had seen with our quantitative real-time PCR. We then went on to look at the protein levels in the plasma, and for this we ran a case-controlled study which included 202 cases, 203 age-matched controls. We also included 15 DCIS patient samples, plasma blood samples, and then 37 patients with benign condition of the breast as well. Now, when we ran the protein levels and did multi-parametric analysis, the best one was achieved with 6 proteins, and the sensitivity, however, was only 65 to 70%, and specificity was around 79 to 85%, which was not sufficient to be used clinically. So we then went on to do cell-free DNA isolation from plasma, and for this we chose 3 of the down-regulated genes, WIF1, DAC2, and SOSDC1. Confirmed that these 3 genes promoter was methylated, and then we went about doing MSPCR after bisulfite conversion, and found that these 3 genes are methylated only in the tumor cell lines but not in lymphocytes. And we use the ratio of intensity of methylated and unmethylated bands were obtained. We then ran all the samples of the case control study. Now, between tumors wherein we clubbed the invasive and non-invasive and controls, which were the quality controls and patients with benign conditions, our sensitivity was 99 to 100% and specificity between 89 to 92%. So what we now plan is to do a large-scale validation with larger number of DCIs and benign conditions of the breast. We are also initiating a collaboration with IIT Madras for the development of a point-of-care device. I need to thank all the patients who have participated in this study and funding agency DST, Department of Science and Technology, and the Institute, my clinical collaborator Dr. Sridevi, pathologist Shirley, epidemiologists Dr. Swaminathan and Rama, and my laboratory colleagues Dr. Sabita Gopal, students Amrita, who had done the case control study based work, and Akhilesh, who's now finished his PhD. So he was working on the running the microarray samples. I'd like to thank you again for your attention, as well as again thank the chairpersons as well as Dr. Sudeep and Dr. Sonanag for giving me this opportunity. Thank you.